the last talk of this session before the coffee break. So Jean Viallet will present us with a talk entitled Unique in Vivo Model for Targeting Drug Candidate Evaluation on Cancer Stem Cells. So he's from Inovotion, and will be also giving a talk on the Chicken Embryo model. Thank you very much, Jean, for accepting our invitation, and the floor is yours.
Okay, thank you for the invitation. Thank you here and all the organizer to the invitation. So I'm Jean Viallet, CEO and founder of the company Inovotion. What we are doing at Inovotion, we develop an in vivo model based on the same systems on Martha previously on the CAM assay. We are doing this since 10 years, and we test the target, make some target validation and drug evaluation. Basically, exactly what we said just previously. We maximize the probability of success and we reduce the time and the cost of in vivo validation. This is true for drug discovery, but this is also true for target validation. And how we do that, we use the chicken egg embryo CAM assay as a pre-screen before to go to the mice between the in vitro and in vivo. Most of the time we are the first proof of concept. In vivo proof of concept, we produce solid go-no-go. The benefits of this model, the model we develop at Inovotion, is that it's very fast. You can get your result in 1 month. We need most of the time with minimal amounts of compound, 1,000 less than with what you will use in mice. We have a very strong reliability because we spent 5 years to standardize our model. I will show you this later. And also because we put a lot of eggs in groups, 15 to 80 eggs per groups. And as Martha says, it's time and cost saving. Recently, last year, we patented the fact that we can use this model for immuno-oncology validation of the compound, including combination with anti-PD-1 and PD-L1. Of course, it's an animal testing it's a in vivo testing with animal testing because of the regulation. And since 5 years at Inovotion, we have carried out more than 550 studies for customers, and we can show now and say now that we have very high predictivity of compound ranking in mice, quite 100%. So very quickly, what is our technology? How we carry out our assay? Just I need to say that we spent 5 years to change completely the CAM assay that was described in the literature. We mastered this CAM assay to make it very robust and highly predictive. So it's not exactly the same as what the other can use. We got the tumor on the CAM, and as the tumor will go in the cancer cell line will go as a tumor. In 10 days, it follows the physiological growth of the embryo. And during this period of 10 days, you can treat the tumor, and you can, at the end, compare the treated tumor to the non-treated tumor, of course, and you can analyze the effect of the treatment on tumor growth, metastatic invasion, and much more other readout I will discuss later. Just to say that if the tumor cell is invasive, you will, if the tumor cell is invasive, you will get a metastatic invasion in the embryo, but also in the lower CAM, and you can find some nodules in all the parts of the embryo. I just want to focus on the fact that it is not a early chick and a young embryo. It's what we call an adult embryo. That means that all the organs at the stage we carry out the experiment, all the organs are there, the liver, the kidney, the heart. but also the metabolism and the immune system is active. And we can find some T cell, B cell, CT cell, NK macrophage in the blood and in the tumor. Here, this is a typical histology of a tumor. It's a real tumor, 3D tumor with vessel inside, immune cell inside, and you can see there is a matrix also, and all the, in fact, I will say the contrary of what was said before, all the microenvironment of the tumor is there and is complete. Even some cancer-associated fibroblasts are there. So we have done many studies for customers since 5 years, and we can test any type of compound, immunotherapy, small molecule, peptide, protein, but we use also standard cell. So for the graft, we use also standard cell line, genetically modified cell line, patient-derived cell line, PDX, but I will show you later CTCs also. We carry out multi-cancer cell line and cancer assay screening, and we can add to the graft some cancer-associated fibroblast, human cancer-associated fibroblasts. We can do any type of treatment, of course, standalone, synergic effect, sequential effect. And there is a long list of readout we can carry out. I just want to focus because it's a cancer stem cell presentation. I just want to focus on the tumor growth, metastatic invasion, but also we can go to the toxicity, teratogenesis, angiogenesis, PK, transcriptomic, immune infiltration histology, and all the readouts you can get in the mice, you can get in the chicken. Just to show you a complete list of what we have, our available model, it's our portfolio for cancer model. We have done, just imagine, 22,000 grafts this year, xenografts this year. On the chicken embryo, we have 50 cancer models, 90 type of cancer. And when we standardize a model, so all those models are standardized, so it's very robust. And when we standardize a model, we standardize also a reference compound. And just I highlight in blue for this presentation all the reference compounds we use and which are currently used in clinical trial for cancer stem cells. So this is an example for standardization of our model. As you can see, we like to have very good dose response for any reference compound. And as you can see, this model is very potent for statistical results. We have very small standard deviation. And so as we put also a large number of eggs, we have very low p-value. This is true at the macroscopic level when we weight the tumor, but this is also true at the transcriptomic level. As you can see, if you compare two replicate, xenograft replicate on the CAM, you can see that there is very few variation between two replicate at the transcriptomic level. I cannot focus on all the readouts, but so I will present a few of them. Tumor growth, where it's an endpoint at Inovotion. So at the end of the experiment, we cut the tumor and we weight tumor and compare the tumor weight between treated or not treated, but could be also between modified cell line and wild type cell line. We can image the tumor and we can, of course, carry out all the other analysis on this. One example of transcriptomic analysis, we set up the transcriptomic analysis with 10,000 human genes on our xenograft. We can compare the expression between treated and not treated tumor or two different modified cell line. In this example, we just compare the xenograft to the cell line culture in vitro, or in vitro and in vivo. And as you can see, there is a large variation between those expressions, and the variation is on different signaling pathway, but it concerns epithelial mesenchyme transition, of course, and the extracellular matrix. So our model is even at the transcriptomic level, it's a real 3D model, it's a real tumor xenograft. Metastatic invasion, of course, is important for cancer stem cells. As I say, if the cell lines are invasive, it will invade the lower cam is here, and also some organ in the embryo. And you can see some foci like this in the lower cam. And at Inovotion, we quantify the metastatic invasion by qPCR using ALU sequence, which are specific for the human cells. And we can compare, of course, the metastatic invasion in routine is on the lower cam, but also in some organ between the treated and not treated xenograft, or between two different cell lines. Just, I want to point out that we were surprised to see that there is an organotrophism of the cell line we use. For example, if you take some non-small cell lung cancer cell line, you will get metastatic invasion in the brain, in the lung, and also in the liver. If you take metastatic prostate cancer, you will find in the bone and the lung. If you take some triple negative breast cancer, you will find some metastasis in the liver and the brain. And interestingly, we were very surprised to see that you can find even a lot of CTC in the blood here. And those CTC, which are in the blood of a chicken embryo, we were able to graft it and to isolate it and graft it and to grow some new tumor. Just one example of what we do with this assay. This was an assay for a customer. We anonymized the result, of course. They came with 4 compounds, and they want to test their compound on lung cancer, and they were testing to see the metastasis in the brain. And what we have seen is that there were 2 reference compounds here, standard of care, 1 and 2. And what we have seen is that none of those compounds were very efficient on the tumor growth, except the standard of care number 2. And when we carry out the analysis on the metastatic invasion in the brain, what we have seen that, in fact, 2 of their compounds were very, very efficient on the metastatic invasion and drastically reduced metastatic invasion. So it was very nice to see how the potential of this assay. Another example here, an academic lab come to test the hypothesis that this microRNA can decrease the tumor invasion when it's overexpressed. So so target validation, a kind of target validation. We graft normal cell line and cell line overexpressing this microRNA. And what we find in the chicken embryos, we observe a drastic reduction of the metastatic invasion with the cell which overexpresses microRNA. And interestingly, 1 year later, they get the result in the mice, and they find exactly the same result on the mice. And this is exactly what we see most of the time. We have a very high predictability of our assay for mice assay. Just I want to finish the last 5 minutes of my presentation on some new assay we set up last year. We were the first one to be able to grow a tumor model derived from patient CTC. It's a very close and very nice collaboration with the Institute of Cancerology of Hospice Civil de Lyon in France. We carry out a clinical assay with 53 patients with lung, breast, and prostate cancer. And we were able to purify by microfluidics the CTCs from those patients. and we were able to graft those CTCs on the choriolentin membrane here. And we were very happy to have a very high number of engraftment, a percentage of engraftment of those CTC on the camas in our assay. And finally, half of the tumor growth and half of we were able to make some very, very nice tumor model for half of the patient, more than half of the patient. Those tumors are very, very flat and very, very consistent, and at the histological levels, it's a real tumor with an histology which is very similar to what we get in mice or in human. And we have carried out, of course, all the biomarker analysis, but I cannot share the results now. But another interesting result, it was that we were able to show that when you take the same patient with different sample, we were able to show that there is the same organotrophic pattern for one patient. And we think now it's correlated. We are confirming now that it's correlated to the pattern in patient. So for the CTCs, we are very convinced that now we have a very good system to amplify the CTCs in tumor models. It's unique. I don't think that anyone in the world has this type of assay in the hand. It's a very fast assay for drug efficacy and resistance. And it's we confirm that the CTCs, the tumor are there is a genetic stability between the tumor and the CTC from the patient. We can carry out biomarker analysis by transcriptomic on those CTCs from the patient and after the tumor growth. And we are still confirming that the metastatic invasion profile is correlated. This assay now is, we want to make it available for clinical assay patient certification, biomarker discovery, of course, companion test assay. So I hope that I convince you that we can use you can use our platform for all the cancer stem cell and therapy projects. That means tumorigenicity, invasiveness, biomarker. I didn't spend too much time, I didn't explain what we can do on drug resistance, but we can discuss about it when you want. And I hope to convince you that Inovotion is a good platform to increase the research result and to save money and save time. I just want to finish to say that our technology is used by many academic labs, and most of them publish in very good journal. And from all those, there is also some patent for all those articles, except perhaps one or two. There is no mice assay, so it's well organized for publication. And of course, I want to thank all the scientific team at Inovotion, and especially Xavier Roussel, which Chloé Prunier, Yann Wang, and Emilien Dosta, which have done most of the work. And of course, I want to thank the HL and the Institute of Cancerology, which is a very nice collaboration for this very nice cooperation and very close collaboration and very exciting results. Thank you. I'm open to all the questions.
Thank you very much, Jean, for a very interesting presentation and quite an applicable technology. So please, anyone who would like to have a question, either send through chat or open up your microphone and camera, and feel free to ask Jean a question. In the meantime, I have one specific question about the CTC assay, which I thought was quite interesting. My question was, it's a twofold question. One is, how long after injection of the circulating tumor cells did it take for the tumors to form?
How long it takes to form?
Mm-hmm.
For the CTC, it's very quick, in fact. It's exactly the same with the cell line. The CTC form very quickly. In 24 hours, you start to have a small tumor, and then the tumor starts to grow.
Okay, and my second question is, for example, if a laboratory, for example, in Spain, for example, my country was interested to do this, the blood is drawn from a patient, and then how long after the blood is drawn from the patient to the time that would have to be sent to you? Is there a window of time for which this is for success rate?
Yes, there's the fact that we need to extract very quickly the CTC, and so the best way to do that is to work with us and the Hospice Civil de Lyon. It's a very large hospital. They have 40,000 new cancer patients every year, so they have all type of cancer, all grade, and so it's more easy to work with us and them to collect the blood than to send the blood from another place. But this is something we are working on.
Right. I mean, for example, if you wanted to monitor patients' response to therapy or do some personalized medicine for a specific patient of yours, this is still a little bit out far.
But this is the next step where we are starting a clinical trial for this. We are on the clinical trial for this to see the productivity of the treatment with our assay, and so this is the next step. We patent this, of course. I will patent this, and this is the next step. And we think about different way to collect the blood and to send the blood to Inovotion.
Okay, thank you. Any additional questions? If not, you can always, throughout the rest of the afternoon session, you can feel free to send. I think we have here, Marina, would you like to ask a question? Can you open up your microphone, please?
No, sorry, it was by chance.
Oh, okay. All right. Then if there are no further questions, then what we will do is close this session.